It is my great pleasure to introduce the first speaker for today, which is Dr. Aditya Nayak. Dr. Nayak started actually as a postdoc here at FAU and is now assistant professor in the Department of Ocean and Mechanical Engineering and a cross hire in the HBOI pillar. And with that, I'm going to turn it over to Aditya.
Thank you, Dr. Scarpinato. Let me just share my screen. So is that good? Yeah. Okay. Hi, everyone. Thank you for coming here to attend this talk. And today I'd like to talk a little bit about some of the research that I do, which is currently heavily focused on using some novel imaging tools to be able to image and understand ocean particles, plankton, and also how these particles and plankton interact with the flow at these small scales. And so before I sort of delve into that, there was a directive from Dr. Scarpinato. We need a brief introduction about ourselves, where we're from, our hobbies, etc. So I grew up in Southwest India in a small town called Ankola, which is in the state of Karnataka. And that's very close to the foothills of the Western Ghats. So for people who are not familiar with it, it's the Western Ghats mountain range is one of the top biodiverse hotspots in the world. So it's a really nice place to grow up in. And there are some of the pictures there of how the monsoon is in my hometown. You know, this is how it is typically during like June, July. Just another brief thing about myself. I really enjoy birding. So my wife and I like to do this a lot in our spare time. So birded a lot across different states in the US as well as India. So here are a few pictures of some really magnificent looking birds that we did. Most of it is Florida and India based, but then a couple of others like in Alaska and Montana. And this is something I really enjoy doing. And as any good birder does, I have a life list. So that's exactly what it sounds like. That basically you keep a list of all the species of birds that you have observed in your life. And I'm currently at 360 plus species and counting. So hopefully that, I can get to like 4 digits sometime. So with that, I'd like to briefly give a broad overview of what I'm going to talk about today. As Dr. Scarpinato said, I got my postdoc at Harbour Branch, but prior to that, I got my bachelor's degree in India at NITK, and then I got a master's and Ph.D. from Johns Hopkins. All my degrees are in mechanical engineering, and I had no idea I would be an oceanographer or I'd be doing something related to ocean as part of my career. I started very late. I do not have any formal training in oceanography. And I got into oceanography only during my PhD dissertation work. So most of my work revolves around developing in situ optical imaging instrumentation and using them, applying them in a variety of scenarios, specifically focusing on characterizing marine particles and also looking at the interactions between particles and their surroundings, specifically the small scale flow physics, right? How two different particles interact with each other, or how a specific particle or plankton responds to a change in flow, and things of the sort. So before I sort of jump into the details of imaging and talk about specific projects, I think it's important to take a step back and just give a brief overview of what I mean when I say ocean particles, right? So typically when people are thinking about the ocean, they're thinking about this vast expanse of water. But in reality, a lot, like every single milliliter of water in the ocean contains thousands of particles, right? And these particles could be many things. It could be suspended sediments or minerals. As you can see in the first image on the top left, it's a suspended sediment plume. The second image shows a diatom, which is a kind of phytoplankton, essentially microscopic plant. So phytoplankton have chloroplasts, which contain chlorophyll. So they're essentially the equivalent of land plants or trees in the ocean. Right. So there are single-celled phytoplankton, or there are colonial phytoplankton, where these single-celled organisms link together to form chains. You also have zooplankton, like you're seeing in the top right and also the bottom right. So these are different zooplankton, which are essentially microscopic animals, for want of a better word. You also have something known as marine snow, which is basically dead and decaying organic matter, which constitutes a significant fraction of what you see in the ocean. And finally, the bane of the oceans currently, microplastics, right? So if you observe the zooplankton on the top right very closely, you can see a lot of blue particles dotted on there. So those are actually microplastic pieces embedded within the organism, right? So that gives you an overall idea of what constitutes of ocean particles. So let's just talk briefly about why ocean particles are important. So they play major roles in very diverse fields. For example, you have suspended sand and sediment. They play a really important role in coastal erosion or sediment transport studies. The ocean as a whole is a big carbon sink. So a lot of anthropogenically generated carbon is absorbed by the ocean. And some of the phytoplankton can fix the carbon and use it during photosynthesis. But then also the sinking particles transport the carbon to the ocean interiors, to the ocean seabed, right? So this particle flux is really critical in trying to sort of look at the ocean carbon budget and consequently relate that to climate change topics. Also, the presence or absence of particles changes how light propagates through the ocean. And so that's also, what is present in the water is very important to optical oceanographers and people who are interested in remote sensing. And finally, a lot of the stuff that we're talking about is phytoplankton, which is the base of the marine food webs, most marine food webs, and so that's very critical to marine ecology, right? And we'll talk a little bit more about phytoplankton. So this is just a collage of different phytoplankton, right? There are thousands and thousands of species of phytoplankton in the ocean. And one little known fact outside of, I guess, oceanographers, or maybe people know it, I don't know, is that phytoplankton are responsible for generating about 30 to 50% of the oxygen that's available to us. So think about it, every second or third breath that you take is basically oxygen generated from not like a tree in a rainforest, but this tiny diatom or some other phytoplankton in the water. And as you can see from here, they have evolved to have this really incredible diversity in shapes and sizes. And there's a lot of interest in understanding why that happened and how, what would be a favorable condition for them to enhance their primary productivity. For example, a lot of ecology questions out there. So with that in mind, and that sort of lays the context of why studying these particles is important. And traditionally, what people have done over the, before the turn of the century, for example, before imaging became a feasible option, is traditionally you just collect water samples, look at them, and try and characterize what's there in the water. Obviously, this has limitations in terms of temporal and spatial resolution. It's very coarse. So imaging, as the imaging technologies developed, it's become a really nice go-to sort of tool for oceanographers to be able to get really high-scale, high-resolution measurements of what's there in the water column, right? So in this talk, I'm just going to focus on one particular tool which we use a lot, and that's a holographic imaging system. So you can think about the holographic imaging system essentially like a three-dimensional microscope, right? So these are two different versions of the holographic imaging system that are shown here. So if you look at the one on the right, you see these two windows. So anything between that is what constitutes the sample volume. So the interesting thing about holography is that you can get the three-dimensional spatial information of all the particles that are present within your sample volume. Right? And you can resolve particles from a few microns to a few centimeters. So it's a really broad size spectrum that you're able to look at. And currently, we have developed a newer version of the holographic imaging system here, which from an NSF-funded project. And so most of the data I'm showing here is going to be from either of these two systems. So some applications of holography, it has found really wide applications in like the medical field, in fluid mechanics, and lately in oceanography. So some of the things that you can do, you can look at the information about particle number. So you can see how many particles are there. You can get information about the particle size, also the information about the particle shape. You can get an idea of the cross-sectional area, the major minor axis length, the aspect ratio, how these particles are oriented in your flow field, right? So you get a three-dimensional spatial structure. You can also uniquely identify these particles. For example, people have used them in atmospheric studies. People have used holographic imaging systems on a plane to look at ice crystals in clouds, right? So it has a really wide variety of applications. In the ocean, people have used it to look at bubbles, plankton, oil droplets. It's a really good tool to image oil. So you can use it for oil spills. You can also look at how particles are aggregating and disrupting, look at predator-prey interaction between different small organisms. Also, you can get velocity distributions. So with that, I would like to just focus some of the work that we're doing here using holography. Unfortunately, I couldn't talk about everything that we're doing. I've just chosen like 3 or 4 topics here. And I think starting off with harmful algal blooms is probably the best thing for this audience. As you know, locally, it's a big problem, right? So a harmful algal bloom, or a HAB, is basically occurs when you have a sudden increase in concentration of a particular planktonic species, and these species could be toxic, right? They could generate toxins, which can either be toxic by ingestion, or in some cases, these toxins can aerosolize, and it can cause respiratory irritation if you breathe them. There are other ways in which these HABs can be harmful without being toxic. They can just be so dense that they can cut off all the sunlight from the from propagating to the bottom. So that can affect benthic communities such as seagrass and corals. It can also just use up all the oxygen in the water and deplete it and lead to a large amount of fish kills. So there are a lot of contributing factors to it, from excess nutrient availability due to, say, agricultural runoffs, favorable physical conditions. So each harmful algal bloom has sort of a unique set of underlying mechanisms which lead to its formation. So people are really interested in quantifying these mechanisms better in order to help mitigate some of these blooms. So we've had success in the past. So working with Tim Moore and so Mike Twardowski and Jim Sullivan, who were part of this initial project. So our lab did a lot of the data processing for this. So the holographic imaging system was deployed in Lake Erie, and where you had a microcystis bloom. So microcystis is a blue-green algae, which is basically the one that is plaguing Lake Okeechobee currently, and also the Indian River Lagoon, both of them in our backyard, right? So it's the same exact species, and the system was slowly profiled through the entire depth of the water column. And so on the right, you see a hologram showing a particular colony of microcystis. So with the data, we had high resolution vertical data. You can look at vertical profiles and how this particular organism is distributed in concentration over depth, right? So that's what you're seeing on the bottom left here. These specific observations, we found that there were 2 dominant sets of cyanobacteria, or these harmful algae, and both of them had variations in their light level preferences, and they occupied different regions of the water column. So this was something which was an interesting finding, because that sort of gives you an idea of the of how of ecology of these organisms, right? So our goal is to pursue this further. The NOAA in the Great Lakes has a team called GLURL, the Great Lakes I think Environmental Research Lab, and we're working with people there to actively looking for more funding to have a permanent installation of the system in Lake Erie. Closer to us, we just wrote a proposal and we were funded from the Florida Fish and Wildlife Commission to use the same technology, that's the same instrument to monitor the red tide, which is another species of harmful algal bloom, which is occurring on the West Coast, right? So it's really far worse there than the situations we have had here in the last few years. So we recently got funded and we're excited to start our work on this project imminently. So I think fortunately there has been no red tide occurrence as of last week. I don't know if it's fortunate or unfortunate in the context of this proposal. We have 1 year to get the data, but there has been no red tide incident so far. So the other people involved here, we're working with a team from USF. Locally at Harbor Branch, we have Tim Moore, Malcolm McFarland, and Lisa Nyman, who's my postdoc, who's also a co-I on the proposal. The second topic I want to go over is, again, related to algal blooms, but these phenomena are specifically known as thin layers. And thin layers are exactly what they sound like. They're enhanced regions of phytoplankton concentration, plankton concentration in the ocean. Horizontally speaking, they can be spread across several tens of kilometers, but vertically they are very restricted. They find like an ideal depth in the water column, and maybe there are a few tens of centimeter to maybe like a meter thick, right? So you can see how it's going to be very easy if you're using traditional sampling methods and sampling every meter within the water column to miss something like this. So imaging, again, plays a very important role here to understand thin layers better. And these are really ecologically important because there are a lot of plankton accumulating at one place. So you can imagine they are a prime food source for higher trophic levels. So I just want to play this video here. And just before that, so this is a video of the raw holograms being recorded. So a lot of stuff is going to be out of focus. But if you look at these elongated particles that you're seeing here, these are diatom chains, individual diatom chains, so basically colonial phytoplankton. And you'll see us that we're slowly profiling to the depth of the water column, and you'll see the depth change here. And as we're doing this, you can see, I'll pause the video for a little bit when we hit this thin layer. So right here, you see a sudden enhanced concentration in this particular particle, particular diatom, right? So we are actually profiling through the thin layer, and we are able to make observations within this region here. And this was data which was collected at East Sound in Washington in the Pacific Northwest. So that's another sort of research thread. A third thing I want to talk about is this is some really nice stuff that my postdoc has been working on recently. This was data collected from a field cruise in the Atlantic Ocean in the Delaware Shelf off the coast of Delaware. And this was a collaborative research effort with the Naval Research Lab. There was multiple different groups with different instrumentation, but we had this holographic imaging system. And I've shown you incidences of plankton previously, phytoplankton, right? So the microscopic plants. Let me show a little bit about the zooplankton. So the zooplanktons you'll see here are mainly ciliates. And so what I'm going to show here is I'm also going to plot the chlorophyll profile alongside the hologram profile. So when we're going down with depth. So the reason to do that is typically chlorophyll is used as a proxy to get phytoplankton biomass in the ocean, because phytoplankton have chlorophyll. So if you if you have a chlorophyll measurement, you usually assume it's coming from phytoplankton, right? So let me play this video, and as we're going down with depth, you see the chlorophyll concentration is pretty stationary, and then it starts increasing. And as it starts increasing, I'll pause a bit here. You see an increase in the number of these smaller organisms, right? So these, I wish I could have zoomed in further and shown that you. So these are these organisms known as ciliates, which are zooplankton, right? So remember, zooplankton, they don't have typically, I mean, they're not an indicator of chlorophyll, right? So because they don't have any chloroplasts. So let me start. So there you go. So this is a ciliate thin layer. You have a bunch of ciliates, but the chlorophyll peak is still increasing dramatically, and it keeps increasing, and then it dies down as the ciliate count goes on. There's a very clear correlation with the ciliates and the chlorophyll. So we found that what's actually happening here is this phenomenon known as kleptoplasty. So literally that stands for stealing of chloroplasts. So basically the ciliates have fed upon all the phytoplankton that are in the general vicinity, not all maybe, but whatever, a lot of phytoplankton, and they are retaining the chloroplasts, right? So they retain the chloroplasts for future use. So if you just looked at the chlorophyll profile without looking at the holograms or without any visual documentation, you would just assume that this is a peak, which is corresponding to phytoplankton, right? So that's how you would look at it. So this is a really interesting observation, and we are writing up a couple of papers based on this data set currently. The final thread I want to just talk about is particle or plankton orientation. So, so the question is, one of the questions we wanted to ask is, you're imaging this in the ocean, you're trying to image it really slowly, so you're looking at things undisturbed. So basically, you're trying to look at particles in the natural environment and how they would be oriented, in the natural environment. So why is particle orientation important? So this is just a brief cartoon which shows that. So if you have a particle vertically oriented as opposed to horizontally oriented, and you have an incident light source, basically sunlight coming from the top, the light scatters very differently when it's oriented in a horizontal position as opposed to a vertical position or some other position, right? So if this is happening on a large scale, then that can really change the optical signature. Secondly, we spoke about colonial diatoms forming these long chains. So if these long chains could essentially be horizontal, right? So what that means is if they could align themselves horizontally, they have a lot of chloroplasts along them. And so they could potentially, and this is just a hypothesis, potentially increase their light capture and enhance their primary productivity. So the two questions, so one is whether particles exhibit preferential orientation, and the second question is how does fine scale flow structure influence particle orientation? And what we found from our field observations, I'm just going to mention it, is yes, emphatically yes. So particles did exhibit non-random orientation in the horizontal direction. That was a function of the aspect ratio of the particle. For example, basically the ratio of the length to the width of the particle. So the higher the aspect ratio, the more probability of this particle being oriented horizontally to the flow direction. So that really was one of the first observations, comprehensive observations of particle orientation in the ocean. We published that, and there we also have more NSF funding to do controlled experiments with different phytoplankton species in the lab to further understand this phenomenon better. And I just wanted to give a brief thing. So this is where the collaborative aspect comes in. I wanted to mention a little bit about more about the collaborations that we have, right? So people who have a computational background might already have thought of this, right? So we have this enormous database. For example, every 10-minute profile from the imaging instrument gives us about 4,000 to 5,000 holograms, right? So now you're doing this multiple stations across different oceans. So we have data from the Gulf of Mexico, Lake Erie, Atlantic Ocean, Pacific Northwest. So we have like millions and millions of holograms. So now the question is, we have barely scratched the surface of looking at some of the data here. The question is, well, you have this vast data set. Now, can you use automated classification means to able to go through this? And I'm grateful to the Division of Research as well as the College of Engineering and iSense. So we got a seed grant to establish a collaboration with Dr. Dimitris Padas and Georgios Sklivanitis at the SEEKS department. And we are currently working with them on developing such an algorithm. So some of the data has already, some of the work has already been done, and Kavita, who's a graduate student at SEEKS, just presented some of the results, and won a place in the best student presentation at IEEE Oceans. So the final slide is basically, I want to thank my research team, which is basically has played a big role, right, in most of what's happening here. So Lisa is my postdoc, Ranjoy is my PhD student, who's working on the orientation work. Lisa's dabbling a little bit with the automated classification and also some of the kleptoclasty data that I showed previously. Paige and Sean have been undergraduate interns over summer who have been really assiduously working on building this label database for automated classification and sifting through thousands of holograms and cropping these individual images. They've put in a lot of effort there. And also I should mention, we have a volunteer in the lab who's also doing the same thing at Harbor Branch. And finally, so this is basically, my work is at the interface of experimental fluid mechanics, physical oceanography, optics, and biological oceanography. I am mainly an experimentalist. I don't have a lot of computational background. But I would definitely love to collaborate with experts in a variety of research fields, especially biologists, because I don't have a biology background, or ecologists, and also people who are interested in computational work. And I also have a link to my lab website. And I guess with that, I can take any questions probably later on once Behnaz is done with it all.
Great. Thank you so much, Aditya. That was excellent. And one thing that I forgot to mention in the beginning is that if you have questions, please use the Q&A button in the bottom of your screen. If you hover your mouse there, you can have a little pop-up and you click on the Q&A button and please type them there. We will go to the questions after both speakers are done. So you would indeed have to indicate who the question is for that you have. If we're running out of time, we're going to ask our speakers to answer the questions offline, and then we'll post them on our website. With that, I'm going to introduce our next speaker, Dr. Behnaz Ghoraani. Dr. Ghoraani is an associate professor in engineering and also an iSense fellow. And her work is primarily in biomedical and speech signal analysis and machine learning classification and so on. I'm going to turn it over to her so she can tell you more about that.
Yes, hi. Thank you so much for the introduction. So you see my screen, right?
Yes, we see it.
Okay, perfect. So thank you so much for inviting me, and thanks for my colleagues for attending my talk today. So let me see. Okay, hold on, sorry. I just don't know why the cursor. Okay, perfect. So I wanted to give a little bit background about myself. So I did my bachelor and then in electrical computer engineering. Basically, I was more into electronic designs, and then I did my internship and then in electronic design, and I have like a showcase of some of the equipment that I designed when I was doing that. So then after while I was working, I did also my master's in electrical engineering, and then after a couple of years I decided to continue and I did my PhD. So basically my bachelor and my master, I did them in Tehran in Iran, and then my PhD I did it in Canada at Ryerson University. And so as I was doing my PhD, I learned more about the medical applications of my algorithms that I was developing as part of my studies. So that was when I decided for my postdoc to go more towards that kind of avenue. And then I did my postdoc in medicine, and then after that I got a faculty position at Rochester Institute of Technology, and I was in the biomedical engineering department for 4 years, and then in 2016 I decided to join FAU, and I've been here since then. So I wanted to share something outside of my work here. So I'm very active with the Institute of Electrical and Electronics Engineering. So I do basically a lot of organizations and activities with them. So I'm the chair of their young professionals, and also I do organize different events. So we have great events planned for our upcoming conference in ICIP, which is going to be about 10 days from now. So if you're interested, so we have great talks and workshops, which are mostly free now because of this virtual situation and the pandemic thing. So we have actually one workshop on how to give a successful presentations, virtually. So I think that would be an interesting workshop to attend. And also, I'm very passionate about women in engineering. So I'm also the associate editor of our IEEE signal processing newsletter. So every month, what I do, I feature one successful woman in signal processing. So like we have great, interviews with these successful female scientists in signal processing. So I also encourage you, if you're interested, to take a look at those. So now more about my research here at FAU. My lab, biomedical signal and image analysis lab. So our goal here is to provide, to help the physicians to provide the right treatment and the right medication for the right person and at the right time. So what we do, our goal is to personalize the treatment or the therapy for the patients. And today, at first, I'm going to focus on our three main projects. And then if you have time, I'm just going to quickly go over some of the other projects just to give you guys an idea about the other type of the work that we do. Just a brief introduction, we do work with our, we see our human body, and the signals that, we can collect from our human body using sensors as a way for our body to talk with us. So we use algorithms to translate that to something that can be used for better diagnosis or better treatment, or even for, prognosis and prevention. So a lot of our work is mainly on this data analytics parts and artificial intelligence parts of the work. So how we can translate that information to something useful for a clinician. And that's why I collaborate closely with, MDs and physicians in different areas in neurology and cardiovascular as well. So the first project I'm going to talk about is on my project on Parkinson's disease. So just a brief, very, very brief description. Parkinson's disease is a chronic neurodegenerative disease that is caused by loss of dopamine in the brain. So you may know about a Parkinson's disease by trauma, but again, it's more into that. It has, difficulty with facial expression, difficulty with gait walking, and then it could also have some problems with, memory, dementia, and depression. So just give you guys some numbers. So 1 in 300 people in the world are basically involved and are affected by Parkinson's disease. And by age of 50, this stat becomes 1 in 80 people. And it has huge burden, economic burden, and also, which is like about $15 billion every year. And then this is expected to double by 2040. Just give you a description of one of the problems that we are focusing on here. So this was about couple of years ago, one of my neurology collaborator, she approached me with this problem that they have patients, and as these patients, they've been medicated, they develop this kind of a side effect that what happens is that they develop this kinetic movement, means that, so they have these involuntary movements. So we've seen it as people like moving their head or their, kind of their arms or the legs. So what she was describing to us was that these patients, when they give their medication to the patient throughout the day, they may feel very, before they take medication, they may feel the symptoms, but as soon as the medication kicks in, they may have the side effect of the medication. And, and then also as the medication is working, all of a sudden the medication can worn off. So there was a lot of challenges for them in terms of what is the right dose and frequency of these medications that the patient has to take. The way that they handle this right now in the clinic is basically by having clinical examinations. The patient comes and they do some, assessment, and also they ask some questions. So the assessments basically shows the physician how the person, the symptoms are at that moment. So it's really not showing the physician how the patient feels at home. And then the interview is like questions like how is your on time, how long your medication lasts, or how long it takes for you to medication kick in. So a lot of these questions, you know, patients have difficulty kind of providing the answer or the, you know, that the physician is looking for. First of all, because of not being very familiar with the terminology that the physicians are using, and also with patient recall. So they may not remember exactly what was the situation over the past 3 months, per se. So there's a lot of adjusting, and it's like a very iterative process for the physician to make these adjustments. And as a result, many patients remain overtreated or undertreated. So the idea is that we can use these IMU sensors, these sensors, you know, that, you know, one can go on the one ankle, and one ankle, one on wrist of the person. So using these two sensors, we can actually develop algorithms that can automate this process and give a report to the physician, that the physician know during that time what was the duration of the time that the medication was working, this, for example, this gray area, or when the medication wasn't working, this red area, or when the medication, you know, had a side effect, this green area. And also about the severity of these symptoms. For example, about tremor, you know, how severe the tremor was, or dyskinesia was, or this UPDRS, which is an assessment or a rating of the severity of the symptoms in general. So with this in mind, we collected data from 24 subjects. It was in a clinic. We had like a home setting environment. It was more simulated home environment. And then we asked the patients to come to the clinic for 4 hours, let's say in this scenario. And then every hour we performed the clinical assessments. And also we asked the patients to do some activity of daily living, for example, like, you know, combing their hair, you know, drinking, eating, or, you know, opening groceries. And so basically we collected this data, and then another one, we did more continuous data collection over 2 hours. And then with this, we had a gold standard of what the physician assessment was, and then we have the sensor data from these patients. So this is just a very, like some of the video that for our data collection. So this is like medication is not working at the beginning, and person walking, drinking, and then we repeated the same thing. Now medication is working, they're walking, and kind of give you an idea about this. So with our, again, I'm not going into the algorithm, but you know what, I'm giving more like just the results. So we did actually, we were able to develop algorithms that we use these two sensors data and then detect, for example, severity of tremor with a very high correlation with what the physician was assessing. And then the important thing about this one is that, for example, here it shows that how closely, you know, this green, which is our model estimated, is matching the blue one, which is what the physician was estimated. And then the beauty of this one is that the patient was doing their activities of daily living. They were just doing whatever they were doing. It wasn't like they were doing exactly the assessment that the physician was performing. You see that this is like the physician performed the assessment, and then after that we do the assessment. So it's like a little bit after this, we make sure that we are able to continuously measure these scales. So that's one big important factor for us, so that we can actually later take these algorithms, this technology to home, and then make the same assessments as the person was just doing, you know, their own daily living activities. So we have also, you know, we are able to measure this kinetic movement. Again, those involuntary movement with very high correlation with what the physician was measuring. And we use, you know, deep learning approach, for example, for this one. What we do, we look at how the, you know, we look at the long-term patterns in this data, and also the short-term patterns in this data, so that we are able to make a very accurate prediction of these motor complications. So we also are able, for example, in this example, we are able to make medication on and off. You know, we know that, for example, during this black time, medication was worn off, and then, you know, and then during this blue time was the medication working. So with this, you know, success that we had in the clinic, so we had several publications, and now we are working to basically to take this in a home environment. So we were successful in a clinical setting in a simulated home environment. Now we are taking this to home so that we can actually make the same assessment and refine our algorithms, that we can make a similar detection for. So I'm collaborating with Mount Sinai and University of Miami to make such a data collection and you know, extension to our work. So another work is we work on management of atrial fibrillation. Just give a brief description about atrial fibrillation. It's affecting, you know, about, you know, 3 to 6 million people in America, and it's associated, you know, you may feel it like as a kind of a some weird feeling in the chest, or you may feel like as, you know, person feels fatigue, or, you know, they have difficulty performing their daily living activities, or, you know, exercising. And and then this is always happening because the disturbance we have in the electrical signals of the heart. But before I go there, I wanted to show you the to remind you that, you know, the mechanical movement of the heart is basically stimulated by the electrical signals in the heart. So as the electrical signals are propagating in the heart, the mechanical movements are happening. So what happens during this atrial fibrillation, there is a disturbance in the electrical signals, that the way that they're propagating in the 2 upper chamber of the heart. And as a result, you know, you may have irregularity of the heartbeats, you know, which you see here. Here you see that very, you know, kind of a nicely, you know, kind of equally spaced heart rhythm in a, you know, normal heartbeat. And then here, this is AFib, which you see that you know, there's like irregular heartbeats. So that's one of the basically ways to detect atrial fibrillation. So it's you know, if this disturbance happens in this 2 upper chamber, then there is a high risk of the person, you know, may die in a few minutes. But when it happens in the, you know, 2 upper chamber, some people may not even feel it. Some people may have those symptoms that I just mentioned. But the problem is that then your heart basically is not functioning in its you know, the highest capacity, basically. So what happens, the blood, kind of these 2 upper chamber, they don't, you know, do their pumping anymore. And they kind of quiver. And as a result of that, you know, it kind of becomes like a bowl of blood. And then it may actually increase the risk of developing blood clots and then stroke. So there are different ways, you know, for treatment. One is that they use medication. Unfortunately, is not very effective, and it has side effects. Sometimes they perform external cardioversion, but again, the AFib can return again. So it hasn't shown to be a permanent solution. And one solution that's been very effective is MACE procedure, which is basically an open heart surgery. And then they insert some cuts inside the atrium, and that interrupts this AFib from happening and returning again. But again, it's a very invasive process, and it's not gonna, they don't perform this process unless the person is going for open heart surgery for any reason, for any other reason, not because of the AFib. The most common approach is this catheter ablation, which, you know, I'm gonna give you some idea what's basically it's really happening. So the hypothesis is that during atrial fibrillation, so these 4 veins that connect this chamber to the lungs, what happens is that as we age or to whatever reason, so some cardiac cells grow inside these veins. And those cardiac cells start, you know, to kind of beat by themselves. And then the electrical signals that they generate come inside this chamber and then disturbs the electrical signals in the heart. So during this procedure, they insert a catheter inside the heart, and they burn the tissues around these veins. And then basically what they do, they electronically isolate, this vein from these, this chamber from these veins. And that's what they are basically stopping AFib from happening and letting the heart to go back to its normal rhythm. So here, this that I show you here is one of our patients. The gray one is basically shows this left atrium of the patient, and then the red dots are where they ablated the patient's heart. But unfortunately, with all these cells that have been destroyed, there is a very high risk of returning AFib. So basically, the patient may actually, some cases, they may leave the hospital, AFib is stopped, terminated, but they may actually experience it again in less than a year. And also in some cases with this procedure, still it may not stop. So the person can actually, just to make sure that, the person doesn't go home with AFib, they may do external cardioversion. So the AFib stops, but again, they know that it's going to come back. So with all this, all these cells destroyed, and then because of these, expensive procedure, with this, the economic burden, and also still patients are suffering. So the idea here is that, so these, as they are, performing this procedure, these catheters go inside the person's heart anyways. So the physicians use these catheters for different applications, to measure electrical signals in the heart. And then, for example, this is they insert this catheter, and these kind of shiny metals are sensors that measure the electrical signals. For example, here for this person, this circle represents one of these catheters, and then these electrical signals that you see is the signal that's measured at one of these, these electrodes that you see here. So the idea here is that these electrical signals are already being collected. They go inside the heart. So why we don't use these signals so that we can help the physician to better identify the sources of these AFibs that are happening. So basically in my postdoc, what I did, I was working in this EP lab. What we observed was that there are some specific patterns inside the heart that, are hypothesized that they are the ones that are causing the AFib not to stop. So just by isolating the left atrium from the veins is not sufficient. So you need to find those sites. And then when those sites are identified, then the physician can actually target those specific sites and burn them and stop AFib from happening. So basically with this way, we wanted to make it a personalized, ablation rather than just driven by the anatomy. So this way we could actually tell the physician exactly this is the site that needs to be burned. But again, we really don't know what's happening inside the person's heart, and then we can't really take these work to at the patient level. So what we did, we first did some simulation studies. So we basically performed computer simulations of cardiac cells, and then we extended to tissue modeling, and then we applied that on the human anatomy, and then use that as our test bed, for our algorithms. So here, this is, for example, it shows that this is, one tissue that we simulated in our, using the approach I mentioned, like it's a 10 by 10 centimeter tissue of cardiac cells. And then, so here, so you see that these electrical signals, as they're basically measuring these electrical signals. So now we developed our algorithms to give us two things. One was that we looked for patterns, and then we can tell the algorithm actually where the source, the direction of the source from the current placement of the catheter. So this is the catheter here, for example. So our algorithms are intelligently telling the physician that this is the direction that the possible source is located. So if you're inside a person's heart, you really want to go to that direction. So we also give the physician a probability of having a source in that area. So we give basically these two metrics. And so with these algorithms, we actually be able to detect these sources in our we've generated so many different computer simulation scenarios that are known to exist in human AFib. And then we were able to detect them with very high accuracy. So here in these graphs, for example, the blue ones are algorithms, which they provide very consistent high accuracy across all these variations of AFib that were simulated. And then these red ones are something that is being used in a clinical setting using a basket catheter, which is a very specific type. So it's not something that is being used in a physician, in a routine therapy. So it's something that the physician has to just send inside the heart just for this application. So what we are saying is that without any basically a specific or a special tool here, with what they basically have in their part of their routine clinical setting, we are able to provide a very high accuracy in detecting those sites. So we also performed, some human experiments. So this is from some of like a patient we had from University of Miami here. So here again, the color represents that in this area, there is a possible source here, and then the arrows are kind of like where it guided the catheter towards this area. And there's another possible source here in this person's atrium. So the next step would be actually performing, this is something done offline, but the next idea would be to perform this in real time so that we can actually, the physician can actually burn the site and then they see that's actually stopping the AFib. So that's where we are right now. Another project I'm going to talk about it very briefly is early detection of Alzheimer's disease. So again, it's not the most common neurodegenerative disease with dementia. So what we are trying to do, we are trying to use our gauge as a way to detect Alzheimer's disease and its early signs as mild cognitive impairment. So here, in the GATE lab here at what we have here, which is now at a division of research right now. So we have these computerized walkway. So as the person is walking in this computerized walkway, we also had some sensors, IMU sensors connected to the person. And this person is walking to this gateway. And then as they do this, basically, their normal walking, and then we ask them to repeat this walking, but now we add, cognitive load, if you may. So for example, we ask the person to say some alphabets, or we ask them to count backwards. So we know that there is a drop in our functional performance as we add some, tasks, some cognitive tasks, but we know that for patients with risk of developing of cognitive impairment, they have a bigger drop in their performance. So we actually use the computerized walkway at this point, and then using only the gauge, we are able to make 78% of detection of healthy from mild cognitive impairment and Alzheimer's disease. And now actually we are using, kind of extending our algorithm. Instead of using the computerized walkway, we are using only the data from these sensors. Now we want to make the prediction without this computerized walkway, be it with our sensors only. And this would be a way for us to take this approach at home. So for example, we can have the person walking and the person walking and then talking on their phone. And then we can use this as an approach to be able to assess the patients in their natural living environment. So there are some other projects, so I'm not going to talk about it, but again, so I collaborate here with Dr. Crystal Wilcox here in psychology. We do develop different novel algorithms for, different kind of way for them to kind of to analyze the FNIRS data. So some of the different research projects we have in the lab, just to give you an idea. But again, I'm going to end it. And so I wanted to thank my students. So it's been, nothing would have been done without their hard work. And also with my collaborators and then sponsors, some National Science Foundations, National Institute of Health, and, Department of, Florida Department of Health, and also seed funding from iSense and College of Engineering and Computer Science. Thank you.
Great, thanks so much, Behnaz. And as a reminder, if you have any questions, please hover your mouse in the bottom. There's a Q&A button. And you can type your questions there. We just have time for just a few questions, and if we have additional questions, we'll ask our speakers to answer those offline and post them on our website. The first question is for Aditya. What is the color-coded map of the ocean? Or what does a color-coded map of the ocean mean?
So if that question is specific to the first of one of the first slides I showed, that corresponds to chlorophyll. So it's like satellite imagery of chlorophyll. So the warmer colors correspond to a higher chlorophyll concentration. So essentially more phytoplankton presence.
Okay, another question. Go ahead. Another question for you, Aditya. How do you collect the face information for your images?
So all the data I've shown you, we've just looked at the intensity portion of the holographic images. So we've not really worked with the face yet. So it's just intensity-based analysis.
The related question here is, can you please explain why you need holography specifically for imaging?
Certainly. So there are a lot of advantages to holography. It's not like we need to specifically use holography, right? I mean, there are multiple other imaging options for looking at oceanic particles. So people have in situ flow cytometers, for example, specifically for ocean particles. The issue with that is the samples that they can process over a certain time is really small. So holography essentially gives you an option to look at three-dimensional distributions, free stream, and you're looking at really large sample volumes as opposed to other mechanisms. Also, because of the 3 dimensions, that also leaves you open to be able to use the data. For example, if you set up your system adequately, you can use like successive holograms to look at how particles change, basically move with the flow to get an idea of the flow distributions. That's something you cannot do with some other mechanisms, right? So if you're really interested in not just looking at the particles, but also interested in characterizing the flow and then tying that with particle flow interactions, I think it's a really useful tool from that standpoint.
So there is a follow-up question to that, Aditya, which is, doesn't holography need face information for 3D imaging?
Not really. So basically, you can use your a numerical reconstruction algorithm to just basically use a Kirchhoff-Fresnel kernel. So basically, use that as a convolution, and then you can get the in focus planes for within your 3-dimensional sample volume. So you don't really need the face information specifically.
Great, thank you. Okay, I'm going to finish with one last question. That one is for Behnaz. What are those specific features that you extract for Alzheimer's detection?
Yeah, so we look at different characteristics of gait, for example, stride length, and then stride time, step time, and then, double support, single support, and all those different gait features.
Thanks. All right, with that, thank you very much to both our speakers this month. We're going to be back with the Research Cafe next month. So thank you very much for joining.
Thank you, everyone.
Thank you.